Alright, so today we're going to look at a monohybrid cross, and mono meaning one trait, hybrid because we're going to cross two different types of parents. So let's just look at leaf shape. It's easy to think about, and in our case we're going to look at crossing parents that have entire leaves versus toothed leaves. And so I've done a little bit of genetics work in this area, and the toothed-leaved plants are recessive to the entire-leaved plants. So entire is dominant to toothed, which is recessive. Okay. And so we have these two parents, and we're going to cross them, and parents are always going to be homozygous for whatever trait they have. And so one parent will be entire-leaved and one parent will be toothed. And entire is dominant, so I'll assign a capital E for it. Tooth is recessive, so I'll use a lowercase. And we know that they're homozygous, so parent 1 has two dominant capital E's, and parent 2 is going to have two lowercase homozygous E's. So we're going to cross these two parents to get our offspring, which are referred to as F1s for first filial generation. Okay, so to set up our Punnett square, we need to figure out what our gametes are. This parent has either a big E or a big E to donate, and let's say that that will be our sperm parent. And then our other parent, which I'll put on the other side of our Punnett square, has a little e or a little e to donate in the eggs. So when I set up my Punnett square, I like to combine columns with rows, always keeping my capital letter first, big E, and the lowercase letter second, little e. Big E, little e, big E, little e, big E, little e. So all of my F1s look the same. They all have the heterozygous phenotype, having one of each type of allele, and because big E is dominant to little e, they will all have the entire phenotype. So this is our genotype, the big E, little e, and our phenotype is what it looks like. Now I'm going to cross two F1s together. So an F1 cross with an F1. They all look the same, so they're all big E, little e, big E, little e, and that is going to give us our F2s, or our second filial generation. So let's set up our Punnett square. Parent 1 has a big E or a little e, and parent 2 has a big E or a little e. Okay, so we put our gametes on the side, and then we draw our Punnett, and we get columns with rows. So big E, big E. I always put my capital letter first. Big E, little e, big E, little a, and two little e's. So my genotypic ratio, I have one big E, two heterozygotes, big E, little e, and one homozygous recessive, little e, little e. So that's my genotypic ratio. And then my phenotypic ratio is what they look like. This will give me the dominant phenotype. This will give me the dominant phenotype. And this will give me the recessive phenotype. So if I add those together, there's 3 entire to 1 tooth, and that's my phenotypic ratio.